makes RNA from the DNA. And so when that's all done, and it zips up again, or like Osmond said, your cosine graph here, you'll have had your initiation site over there, and then your termination site. And the way the enzyme knows to go there and stop there is because of some very specific series of like all of the 4 nitrogenous bases. And it'll just recognize those and bind to those and start moving along. You don't need to know what that code is, and I don't know. I don't think Osmond knows. AUG. AUG? Hush. So, yeah.
I think it's methionine.
Hush.
Alright.
So it'll jump on there and it'll just start binding. Yeah.
Is there a certain direction it goes?
Yes. So it'll always attach to just one strand. The other one's sort of like backup memory. And like I mentioned earlier, you're going to have your 3 on one end and your 5 on one end, and that makes sort of an arrow going from low to high. So this will travel in that same direction, going up the DNA strand. Is that right, Osmond?
Yeah.
Okay. So this goes up, and this other one goes down. It's the exact negative. And the funny thing is that with your messenger RNA, is you're going to have a negative of your positive. So the most important bit that goes in and starts coding those proteins is actually a negative of the instructions. So you have your RNA, and it travels out of the nuclear membrane, goes out of the nucleus, and does anyone who's taken biology know what structure makes proteins when it gets an RNA code? Sir? Ribosomes, exactly right. So this is your messenger RNA, or mRNA, and it'll go out and find a ribosome, rRNA, and if you remember, oh yeah, these will be just kind of dotted around, and also on the big Golgi bodies, and also rough endoplasmic reticulum.
Yeah.
So you'll have these just kind of dotted around. There's so many of them because the cell always needs to be ready to start making proteins. And so this mRNA will feed into one of these massive spewing coils that's actually just made of different RNA. And this acts kind of like an anvil for the transfer RNA. And it looks kind of like that. It's pretty wonky. And so this bit here, I'll make a zoomed in bit in just a second. This will attach the mRNA, and you'll have an amino acid here, and it'll start lining those up, and you'll start getting these amino acids forming together, matched up against the mRNA. And they'll break off from the transfer RNA, or tRNA, and they will start making peptide bonds with each other. And peptide bonds are a lot stronger than the ester bonds that form between the amino acid and the transfer RNA. And so you'll get a stable protein starting to form. And so what will happen is you'll have your mRNA feed in here, and this ribosome will just start munching along, and it'll spew out your protein. And they'll just kind of link together with the peptide bonds. So why do you think this is called transfer RNA? What's it transferring? Yes? Sort of. You're thinking more along the lines of the mRNA, but it does have a code on there that matches up. Yes?
It transfers the amino acids.
Exactly right. This transfer RNA, or tRNA, will transfer the amino acids there, and it holds it together. Now, does anyone mind if I erase this?
Okay. Right before you do.
Actually, I'll erase this.
All right. So out of that ribosome comes amino acids, right? And Jack just mentioned they form peptide bonds to each other. So multiple amino acids with peptide bonds in between them is called a polypeptide. Okay? Poly meaning multiple, and peptide indicating which bond is used to join them. Polypeptides can be proteins themselves or can form together to make proteins. So that's kind of how your protein starts to develop.
Okay. Now, you remember when I said that RNA will sometimes make loops with itself and sometimes make straight passages? Well, tRNA really folds in on itself, and it will make this wonky structure. Let's use orange. I haven't used orange yet. Down here, it'll make an anticodon. So the bottom of this, there will be 3 of all of these nitrogenous bases that would pair up with each other. And these 3 bind to the RNAs. And this is a negative of the DNA. This has the same bases as on RNA as DNA, with the exception of where thymine was, there's now uracil. So these will attach together, and there's an amino acid way up there. Let's say it's alanine. In the future, we'll cover proteins and amino acids. For now, you just need to know that there is an amino acid there. And going that way. So that's the 5 side, that's the 3 side, 3 side, 5 side. That you will need to know. This, the way I remember it, is there's a weak bond here, so it's on the lower number. And there's an ester bond there. And once they all start coating up, like this, you'll get a bunch of different amino acids grouped together. And what is a long group of amino acids called? There's a couple different names. What? Polypeptide?
Yeah.
Or, that is correct, polypeptide. Protein. Right. Proteins are made entirely from amino acid chains. And so, with the ribosome going through the mRNA, it makes these chains. So that, oh, by the way, when I said it makes, RNA makes some really, really wonky shapes when it folds up, this is one strand of RNA, and it's incredibly long. It's folded up on itself, and if you mapped it out, it looks like some subway planner just took goo and splattered it down and made lines. If you ever look it up, it's really cool. That's all for RNA and DNA. Osman, if you don't mind, I'm going to go through onto how these peptide binds actually form and why they're stronger than the ester bonds that bind them.
Okay.
And then I'm done. Does anyone have any questions so far on translation? By the way, what was happening here is translation. Forgot to label that. So you take this instruction and translate it into the different code of the peptide bonds. Whereas transcription, you're taking the DNA code and copying it, transcribing it into RNA.
So that's really important to know, the differences between transcription and translation. So transcription is from DNA to RNA, and what's translation from?
RNA to amino acid.
What? What kind of RNA?
Right, messenger RNA.
Right.
What are the other 2 of 3 types of RNA that exist?
tRNA.
tRNA, transfer, and
rRNA.
rRNA, ribosomal RNA, which is found in ribosomes, which then themselves are on an endoplasmic reticulum and Golgi bodies.
Yeah.
So wait, this is really
Louder, talk louder, talk louder.
this isn't relevant to like translation. Like,
Yes.
you said here that how long are DNA strands? Like 6 feet?
No, like
DNA strands can be extremely long. They're just all coiled up on each other. But because DNA is so stable, RNA tends to break apart after a while, so it's not used for storage except for viruses. But DNA is really stable, and if you stretched out all the DNA in your body, you could circle the Earth. It's pretty long. Question.
Why are some ribosomes on the endoplasmic reticulum and some are not?
Because you need to be able to make them in different places. Most of the time, because the endoplasmic reticulum is kind of enveloping the nucleus, it'll go there. At other times, you'll need to create proteins in different places. And so you do have free-floating ribosomes sometimes that'll catch the extra stuff, and ones in Golgi bodies because those move around. Is that right, Osman?
Yeah, that's correct. Smalley, what was your question again? You never really finished it.
Mine?
Yeah.
Oh, no, I was looking at.
Alright, so we're not good enough for you, I guess?
Yeah. No,
Alright.
no, not that. No, I would, no, because he said they're really long. I just wanted to know, like a rough estimate.
Oh, like, they vary in size from like 1 foot to maybe like 50
So
feet.
I think
maybe.
he might not, but Osman will probably cover later how this will make like a really massive thing.
Yeah.
Yeah.
Okay.
Wait, what did you say?
What time do you have to go?
What are you saying?
No, because he said that each pen was like a mile long.
Yeah, I thought it would be something.
They can be that long. It's really all it is. DNA's super stable.
Guys, listen to Osman.
DNA's super stable. So like Jack said, you have these base pairs that just connect, and it's so thin and so small, they just coil around and around, and like form a really small condensed structure, but still have a long continuous strand. You know. So they don't have like RNA, where like Jack said, RNA does wind around and it bonds with itself in different locations, so it can break apart. But DNA is pretty nice to just go along by itself for a long time.
Thank you. One disappointing thing. Anyone here watched Jurassic Park, the movie? Raise your hand. So, you remember the bit where they're talking about how they went into amber, and they picked out a mosquito that had sucked the blood of a dinosaur, took out the DNA, and then made it into a dinosaur?
Yes.
That doesn't actually work. And we said that DNA is super stable, but it's not perfectly stable. And over time, I believe it was cytosine, tends to decay and turn into thymine, and you can't really tell which is which and which is actually supposed to be there. So over time, the DNA will break apart and start changing on itself. So unfortunately, we can't make anything really older than a woolly mammoth. We have made woolly mammoths. It's pretty cool.
Actually, like, part of it.
Now, this last thing I'm going to cover are 2 amino acids. And if you remember, what is the difference between these 2? Like, what makes one amino acid, the whole thing, different from another amino acid?
Well, one's nice.
Shout it out.
There's like 20, right?
Yeah, that's correct. There are 20 different amino acids. But what is the difference?
The electron.
Not the electrons.
The different codes, right? The different codes.
The arrangement of the nitrogenous bases.
No, there's not nitrogenous bases
Or-
within these. Those are in
One is
DNA.
mean and one is nice. I'm kidding, by the way.
That was a terrible pun.
I know.
So the
alright.
difference, if you remember from last week, I only briefly touched on this, is that this side group is going to be different. So you have an amine group over here, and you have a carboxyl group here, and a carbon and a hydrogen. This bonds to some other molecule. It could be as simple as a single hydrogen atom for one of them, or 12 atoms for another.
This is really important.
Yeah, this is kind of imperative for anything protein
You
related.
might need to write this down. 'Cause what does an amino acid consist of, right?
And if you go, I think I was looking at Stanford's website last night, and they go through in detail about how some are ionic and therefore hydrophilic. Water likes them. I don't know what you're doing. Okay. Write it in a darker color.
You guys read this?
So, yeah. So the different amino types, there are charged, positive and negative, ionic amino acids, and those will be hydrophilic. Some will be entirely nonpolar. There's no difference from one end to the other. And they are hydrophobic. Water likes polar things. And there are some that are just polar within it. It's not ionic. There's no net gain or loss of electrons. But if you look from one side to the other, electrons tend to cluster in different places. And so they'll be polar, and water likes polar things, and it'll come up. So when these react, what's going to happen is, you see, did I draw that wrong? I did not draw that wrong. You're good.